So the paper we're going to talk about today, it's kind of elderly now, but the same approach can be used with genomic sequences or partial genome sequences as was used in this paper using this fragment of DNA. What they did was they cloned a large chunk of DNA from an organism they were interested in, SAR86. So SAR86 was one of these collection of organisms that was first identified in the Sargasso Sea. That's where the SAR comes from. Clone 86. And organisms in this group are really, really common in the ocean. And so the question is, okay, they're abundant. They're gamma proteobacteria. What are they doing? How are they contributing to the ecosystem? If they're that numerous, they've got to be important. And so the question is, what are they doing? And their approach was to fish out a fragment of DNA that they knew was SAR86 because they had that fragment of DNA has a ribosomal RNA in it. That's the phylogenetic information. And then sequence genes around it, so other genes on the same piece of DNA to see what might happen. Yeah.
When you say fished, you mean?
I mean extract that. Yeah. So what they did was and it's described here a little bit. They isolated bacteria from the ocean, filtered them out. So they have a pre-filter of I think 20 microns, and then a post-filter of 0.45 microns, and they collect everything that comes out on that filter, extract DNA, and then they go through this cosmic cloning thing. And I'm not going to bore you with how cosmids work. Nobody does cosmic cloning anymore. It's really a pretty amazing process, but it's a lot of work. The advantage of it is when you do a typical cloning, like in bit 410 or something like that, you're cloning a couple of kb fragment. In this case, you're cloning into lambda phage, and you're cloning pieces up to 150 kb, so really big chunks of DNA. They did this. Their average clone size was 100 kb in length, and then they screened them for the presence of SAR86 ribosomal RNA. And so what they actually did was they made a probe that was specific for the SAR86 ribosomal RNA, and they probed a bunch of these cosmids, and they found one that had the SAR86 ribosomal RNA in it. And so now they've got, in this particular case, this clone was EVAC31A08, contains a 130 kb fragment of DNA. It's got the SAR86 ribosomal RNA in it. So they sequenced the whole thing to see what else was there, hoping that they might find something on there that would give them a clue as to what this organism could do. Now that might seem like a shot in the dark, right? I mean, if you've got one gene, the genome of this thing is going to be probably 2 to 4 kb, 2 to 4 million base pairs. What are the chances that a 130 kb fragment has something on it? Actually pretty good, because that represents about 20% of the genome of an average oceanic organism. And typically, metabolic pathways contain more than one gene, right? And if those genes are scattered around, the chance of one component of that genetic pathway being on that 130 kb fragment, the chances aren't so bad. One protein coding gene is typically about a kb. Of course, it varies dramatically, but that's the average. And so that means you've got approximately 130 genes on that piece. Chances of finding one that gives you a clue about what the organism can do are pretty good. But I have to say they got lucky beyond any reasonable expectation. Yeah?
This organism was unculturable, right?
Was what?
I assume it was unculturable.
Yeah, unculturable. You can click on this link to the NCBI graphic browser site for this species. You can look at all the genes that are on there. It's pretty interesting. But they got really lucky, because one of the genes on this thing showed up as a homolog to opsin. You guys know what opsin is? It's the stuff in your eyes, right, that lets you see. It turns out that there are 3 kinds of opsins. The one you're most familiar with is the traditional opsin. These are proteins found in metazoans, in animals. They have a cofactor called retinal. This is a small molecule, and when it absorbs light, there's a cis bond that switches to trans, and the result is that it's a 4-barrel. I'll show you. I think I've got a picture of it here. It's a 7-barrel transmembrane protein. Those 7 barrels sits in a ring, and so it's like a pore in the membrane. And the retinal sits right in the middle and blocks that pore. When it absorbs light, the conformational change of that retinal opens up, and now calcium ions can flow right through that pore. And so when you've got a retina cell, a rod or a cone, when it absorbs that light, it opens up, calcium flows in, and it starts an action potential. Those cells are essentially nerve cells, right? And you start an action potential, and that's propagated through all the layers of the retina and ultimately to your brain, and you see something. That's the traditional opsin. There's another kind of opsin in archaea called Bacterial Rhodopsin. And I don't have anything specific to that. These are in the halobacteria. And it's the light-driven proton pump that we mentioned briefly earlier. And so in this case, when retinal absorbs that photon, it doesn't open a channel, it boots out a proton. And then it resets, and then it can do that over and over and over again. So as it absorbs more and more light, it pumps protons out of the cell and creates a proton gradient that you can use to make energy. So it's a very simple form of phototrophy. It's just a light-driven proton pump to pump up the gradient so that you can make ATP with ATPase in the traditional fashion. There's a third kind of opsin. These are sensory opsins. These are the proteins that all kinds of organisms use to detect light for a variety of purposes. So there are sensory opsins like this in your pineal gland that detect the light, believe it or not, in your brain, the light that filters through, and it's how your circadian rhythm is set. This is why you get jet lag, one of the reasons you get jet lag. There are lots of other reasons. Fungi use them. When you look at a colony of a fungus, it's not just a big homogeneous colony, right? It's in rings. Why is that? Because the organisms grow on a daily cycle. Just like the rings of a tree, those rings are daily rings of growth. Fungi grow with a circadian rhythm. Phototrophs of all kinds need to detect light for a variety of purposes. So for example, when we talked about the cyanobacteria, we talked about how some of them separate nitrogen fixation and phototrophy on a diurnal cycle. They'll make energy during the day with light, and they'll fix nitrogen at night when there's no oxygen being generated to inhibit nitrogenase. How do they know whether it's day or night? They have these sensory opsins. Phototrophic organisms generally use this technique because the photosynthetic apparatus is expensive. You've got to make a ton of protein, a bunch of cofactors. You've got to know that it's going to be worthwhile. And you know it's going to be worthwhile if there's light. And so typically you have a sensory opsin, and when it detects sufficient levels of light, that turns on all the genes for making all the photosynthetic apparatus. And so here we have an organism, a gamma proteobacterium in the ocean with opsins. The opsins had never been seen in bacteria before. And so the question is, well, the hypothesis is that opsin has something to do with the lifestyle of this organism in the ocean. So the first question is, anytime you see a gene in a genome and you think it might be interesting, the first question you're going to ask is, okay, am I right? Is this really a gene, in this case, for a functional opsin? This tree, by the way, is showing you where it trees out amongst the gamma proteobacteria. It's not treeing out, for example, with the traditional phototrophs like Rhodobacter. So the first question is, okay, if it's a real opsin, let's see if we can figure out where it goes based on a phylogenetic tree. So here, this is a phylogenetic tree of opsins. So these are opsin sequences all aligned, and including this thing they're calling Proteorhodopsin for proteobacterial opsin. And yeah,
So are ours the Rhodopsin?
no, this is it right here.
No, that human one.
Where is the human one? I don't think the human one's on here.
So is
But here's a sensory opsin right here.
Is Rhodopsin another kind of opsin?
Yeah, so I'm going to talk about that in just a moment.
So they're full.
Well, yes, but it's actually a modified version. So this is the phylogenetic tree of various opsins. Most of these are from halophiles. Here's a sensory opsin from Neurospora. The bacterial rhodopsins are proton pumps. The sensory opsins here are these ones. The halorhodopsins are a modified version, which are specifically found in halophiles that are light-driven chloride pumps. So these organisms live in salt lakes, and salt flows into the cell. That's okay. These organisms have highly ionic cytoplasm. That's part of their adaptation to high salt. But chloride's a terrible anion. It's really not very biologically friendly. And so they would rather have something like glutamate, some organic anion. And so they have these light-driven chloride pumps that are like bilge pumps, pumping chloride out of the cell as it leaks in. Anyway, they included the Proteorhodopsin in here, and it looks like it might be a sensory opsin, but the bootstrap value here isn't very good, and the result is they really don't know from this data what kind of an opsin this might be. There's no clear answer to this. They didn't include visual opsins here because those proteins are so different in sequence that the alignments aren't really reliable. And so, unfortunately, all you can say from this tree is that it's probably not a chloride pump, probably not a sensory opsin, well, probably not a eukaryotic sensory opsin, but otherwise, don't know. Given that, you might want to ask the question, okay, is this thing really a functional opsin at all? And so they did a secondary structure prediction of their sequence, and it falls into the nice barrel structure that you might have predicted. And when you, because this protein has been studied to death in halophilic archaea, you know what all the essential amino acid residues are in here for binding that retinal. And they're all present in this sequence. And so this thing looks like it really ought to be functional and not some pseudogene or something like that. So does it work? So they take this gene, they cloned it into E. coli and expressed it. This is a cell suspension expressing this protein. And then you add retinal. Retinal is a colorless cofactor until it binds the protein. And when it sits on the protein, the change in the electronic environment primes it for light absorption. How do you detect light absorption? It's color, right? So you add retinal, and it turns red. Great. This tells you a couple of things. One, that protein probably really is functional because it combined retinal, and it can do it in that E. coli suspension. It's probably in the membrane of E. coli and binding retinal in what would appear to be a reasonable way. And so that's shown here with the absorption. It has a very nice absorption at 520 nanometers, right where you would expect it to be. So what's it doing? The easy experiment to do is to see if it can pump protons, right? And so here's that E. coli suspension. This is suspension, not, suspension. And so not expressing proteorhodopsin, no retinal added. And so you have it in the dark and you turn the light on, and actually it gets a little more alkaline on the outside. So this is pH here on this axis. Certainly you're not pumping protons out and making the outside solution more acidic, which is what would happen. Proteorhodopsin by itself, nothing different. Retinal by itself, nothing different. But in cell suspensions expressing proteorhodopsin with where you've added retinal, now you turn the lights on and the pH outside the cells plummets. And by plummets, I mean 0.02, right? I mean, it's not a lot, but it's clear. So this thing looks like it might be pumping protons. They have a completely independent way of showing this, where they take, these are right side out vesicles. So they take E. coli, they sonicate it, and they make vesicles of them. And then they add this stuff called TPP, which is it's an uncoupler. It can float through the membranes, but it's positively charged. So what happens then is, if those vesicles pump protons out, then in order to maintain, because you've got an uncoupler, you're not going to generate a positive charge on the outside. Every proton that goes out drives a TPP in. And these are radioactive, so you can measure that very carefully. You spin the cells down and count the radioactivity in the pellet. And when you shine light on it, sure enough, TPP accumulates inside the vesicles. So this stuff really does look like it's pumping protons. Is there any other way you can measure this? There's a big, big difference between sensory or optical opsins and pump opsins. If you're a sensory opsin, when you detect that light, the longer that channel stays open, the stronger the signal is, right? Imagine increasing that open time tenfold, that's 10 times the signal. It's a little bit like a gain knob on an electronic, I don't know, a speaker or something. A pump opsin, though, if you stay open longer, you get no more energy out of it, right? You pumped one proton out, that's all you're going to get. Every cycle pumps a proton or a chloride or whatever you're pumping. And so you'd really like that to reset as quickly as possible so that you can absorb another photon and boot another ion out. And so pump opsins recycle very quickly. And sensory opsins recycle very slowly. And so what about this one? So here you're measuring these vesicles. Let's see, are these vesicles? No, these are suspensions of E. coli in this case, in which you've got proteorhodopsin being expressed, in which you've added retinal. And these are millisecond time points here. And they're measuring at 3 different wavelengths after you hit it with a laser. Each of these wavelengths then is measuring a different state of the proteorhodopsin retinal complex. 520, well, this is 500 here, but this is basically the red color that you see in that tube. And so the question is, when you hit it, you see a change in absorption because it switches to the open conformation. And then you're looking to see how long it takes to reset. And so here's the standard absorption. You hit it with the laser, the absorption at 500 nanometers plummets, and then it recycles with a half-life of about, 15 milliseconds. This is the open complex here, and then this is a transient intermediate there. And I'm not going to go through this graph. This is following the same kind of stuff. "15 milliseconds is really fast. You would expect it to be about 20-fold slower if this were a sensory opsin. So this thing really does seem to be a light-driven proton pump in a gamma proteobacterium, not in a halophilic archaeon, but in a gamma proteobacterium. Nobody ever knew this existed. This organism represents, something like, I don't know, 10% overall of the biomass in the surface of the ocean. And it's making energy from light, and we had not a clue. Now, that doesn't mean it's photosynthetic. It does not mean that it's a primary producer, because you don't know from this data whether it can fix carbon. Now, how would you know whether or not this thing was autotrophic based on the data you have, that is this giant chunk of genome? What would be some clues that it could fix carbon? I already gave you the answer to this. What protein would tell you this is an autotroph? Yeah, but is there a specific protein? Rubisco,
Yeah.
right? If it can do the Calvin cycle, it can fix carbon, and if it can make energy from light, and if it can fix carbon, it's an autotroph. If it had proteins for the hydroxypropionate pathway that chloroflexi and some archaea have, that would tell you. If it had proteins for the Wood reaction, then you know that it fixed carbon. The problem is, in data they've done since then, they know it has a complete Calvin cycle, a complete Krebs cycle, TCA cycle. But that doesn't tell you whether or not it can fix carbon, right? Because a lot of organisms have a complete TCA cycle, but can only run it forward, can't run it backwards. In fact, there's no evidence that this organism can fix carbon. They've done the experiments since then and can find no evidence that it can fix carbon. In other words, this organism is a photochemotroph. It can make all the energy it wants from light, but it needs organic material for carbon. That is, it is not a photoautotroph, it's just a phototroph. It's not a primary producer. Nevertheless, that's a bunch of energy added to the ecosystem through this process. And it's probably using that energy, obviously for the basic energy needs of the cell, but imagine, for example, that it can use that energy for really energy-intensive things like motility or nitrogen fixation. Nitrogen fixation will be a big one. Since then, people have pulled a bunch of sequences out of the ocean. Genome sequences, just metagenomic sequences. It turns out that this gene, this Proteorhodopsin, is found in lots of organisms in the ocean from all phylogenetic groups, not just gamma proteobacteria, but beta and alpha proteobacteria from the Bacteroides group, from gram-positives, from all kinds of organisms. This gene, well, I can prove it. All that was needed to make E. coli a photoheterotroph was the presence of one gene and a vitamin, retinal. So think about that. Complete change. Here's an E. coli that can get its energy from light. And it does that based on the transfer of one gene into its genetic complement. In this case, a recombinant plasmid. But you can imagine this happening in the environment too, right? You bind and pull in some DNA, and it happens to be from SAR86, and you've got this retinal chunk of DNA in. It ends up in your genome. If it makes a little bit of it, now you can get some retinal from the environment, then now you're making energy from light, and you're going to keep that. This gene seems to float around all over the microbial ecosystem, a little bit like the NIF genes that are readily transferred, because all you need is a collection of half a dozen genes in a big operon to make you a nitrogen fixer, and that's a big deal. Yeah.
Would you assume that this organism synthesized its own retinal or got it from the environment?
There's SAR86, the genome has been determined since then for a couple of members of this group, and none of them have the genes for retinal synthesis. And so presumably they're getting it from the environment. Something's got to be producing it, and what that is unknown. But And that seems like a real problem, but retinol production is hard. There's a lot of genes involved. So yeah, it'd be nice if they could make it, but all organisms have cofactors they can't make. We call them vitamins, right? And we have them in our diet. These organisms have to pull them out from the environment, but they don't need very much. So it seems to work for them. An interesting thing about this protein is you might imagine, so this particular E. coli, for example, how well do you think it could make energy 150 feet below the surface of the ocean? Using this system. The issue is this is red. Not much light, red light, goes through the ocean water. Once you get down to 100 feet, there really isn't very much red light left over. This is why underwater pictures always look blue, right? So this wouldn't be very good for a deep water organism. It would only be useful for an organism that lives right on the surface where these samples were taken. It turns out that if you look at SAR-86 organisms from different depths of the water, the Proteorhodopsins they have different sequences in here, and it tunes the absorption of the retinal to the kind of wavelengths that they're going to get at that depth. And so you get blue ones in deep water and red ones in shallow water. Yeah?
Why not blue ones?
So that's a good question. Presumably competition, but I don't know the answer to that. This is an interesting quandary because these organisms have to be pretty mobile, right? That takes a bunch of energy. Staying at a particular depth is an interesting problem. It's also true, by the way, that these organisms go up and down in the water column on a daily cycle. They're near the surface at night, and they go deeper during the day, and so forth.
Is it possible that you could have multiple systems?
That's a really good question that I don't know the answer to. So you could imagine having more than one retina or more than one Proteorhodopsin to absorb different wavelengths. But I'm not aware of any organism that has them. But most of the results, most of the information we have about here is from metagenomic data, and you might not know whether more than one of these is in the same organism or not. There might be other ways, of course, to tune these other than the sequence of the Proteorhodopsin. There might be ways to tune them with different lipid compositions, for example, in the membranes.
So one gene family, I'll say, could
In theory.
expand.
Right. And can you give me an example of how you know that's true? How many colors do you see on the screen up there?
I was just thinking,
Black, white,
you don't see the color blue.
The reality is we have 3 of these, right? Well, except for some of you ladies may have 4. There's some fraction of women have 4 different options. And those three different absorption spectra give you all of the color vision you have. And so you could easily imagine that other organisms might have different versions of these so that they could absorb wavelengths at all wavelengths in the visible spectrum. I'm not aware of any of those. So, to conclude, here we have a case where on a single piece of DNA you have phylogenetic information in the form of ribosomal RNA that tells you what the organism is. And you have genetic information about a metabolic process that tells you what it can do. And by being on the same piece of DNA, you know it's in the same organism, and so you've linked the phylotype to the phenotype. On Friday, we'll do this workshop. I'm going to commit to doing that. So instead of this novel antibiotic thing, we're going to do a workshop on the RDP. Particularly useful for those of you who are having some trouble. I've been getting a flow of emails about this. And then next Friday, I'll leave it a little bit open about what we're going to do until I get some more suggestions. I really do like this phylogenetic paper because it, at the very beginning of this class, I argued that all the stuff you need to rethink, right? It'd be cool to do that again with this tree and make you rethink all the stuff you've learned in this class as well. But this pathogen sounds cool too. So, John Luke, what's the name of this organism? Elizabethy, something like that, yeah. All right, so give me your opinions, and I'll see you then on Friday. We'll talk about genomes next week.